When we look at ribose sugars, a pentose, remember this particular type of sugar is called a pentose. And the pentose has 5 carbons. So here's 1, 2, 3, 4, and 5. 5 carbons. 1, 2, 3, 4, and 5. This particular type of pentose is called a ribose. Now there is a difference between this ribose, this 5 carbon sugar, and this deoxyribose. Can you tell me where the oxygen is missing? Well, hopefully you notice that at this position, this ribose sugar has an oxygen, where at this position, this pentose does not. So this is a deoxyribose, D meaning not, right? And oxy, of course, meaning oxygen, and the ribose sugar being the pentose, the 5 carbon ring structure that's made of carbon, hydrogen, and oxygen. Also remember that since it is a sugar, we have C blank H blank O. In this case, if we know we have 5 carbons, then hopefully you know by now that it's 10 hydrogens and 5 oxygens. And so you can count them yourselves, but because we know the general formula for sugar, we know that this ribose has C5H10O5. Except, of course, that deoxyribose is an exception. It's missing that oxygen, isn't it? So it doesn't follow this general formula. But the ribose version, the one with the oxygen, does. You'll notice you can count yourself. There's 5 carbons. There's 5 oxygens. And there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 hydrogens. All right. So now let's also remember that when you're numbering the carbons, when you're trying to label the carbons so that we can all have reference points for our discussion about DNA and RNA, you have to consider which carbon's which. So if we start like any clock with a 0 point, this being our 0 point, then this is carbon 1, and we usually put a prime on it. Prime meaning that little apostrophe. Here's 1 prime. The second carbon is a 2 prime. Here's your 3 prime. There's an important 3 prime. 4. And then, of course, your 5 prime. The ones that we're really interested in, the ones that we really need to worry about, is our 1 prime, our 3 prime, our 1 prime, our 3 prime, and our 5 prime. You'll see why the 1 prime is important soon. The 3 prime, of course, is where we make a connection on the phosphate sugar backbone, and the 5 prime is the other important connection. The 2 prime is important in that it allows us to identify the ribose versus the deoxyribose. This missing oxygen here at the 2 prime is what determines whether it's deoxyribose or ribose. Now when we look at DNA, we see a phosphate sugar backbone. You'll see that there's 2 strands. Let's take a look at this DNA molecule. 2 strands, 1 strand going this way, and 1 strand going this way. please notice that where this strand starts here at 3', it ends down here at 5', and where this one starts at 5', it ends at 3'. DNA is directional, and we'll look at that again in a little while. What do we mean by 3' and 5'? Remember, this is the prime 1, there's 2, this is 3, this is the 3', and 4 is there, and 5 is here. So this 5' is where we make connections between the monomers in the polymer of DNA. Let's take a quick look, close-up look at this. If we number these again, this being your first, here's your second, there's your third, 3', this is your 5' carbon here. This little dot right there. That 5' is connected to the oxygen of the phosphate, which is connected to the 3' on this end. So you see the connection between the 5' and the 3'. 5' of one ribose or deoxyribose sugar. Notice there's no oxygen here, right? The 3' of the 5' of this sugar is connected to the 3' of this sugar. This carbon here is connected to this carbon here by this phosphate group, which is then, of course, has on this end, on this 5', you have another phosphate. Now we stop the connection here just to look at it, but this would continue on for a very long time. So when we look at it, when we zoom out, we would see here's a phosphate, 3' of 1 is connected to the 5', the 5' of 1 is connected to the 3' of the other through this phosphate group, which would be right here in between them. This phosphate group is represented by this blue strip, this blue line you see here. So 3' or 5' of one ribose sugar, deoxyribose sugar connected to the 5' of the other ribose, or 5' of one ribose sugar connected to the 3' of the other ribose sugar by a phosphate in between. The twisting geometry, the helix, the alpha helix twist in the molecule comes from the geometry of this construction. So when we have this turn, these turnings, this is the geometry that's forced by the shape of the molecules themselves. So you have these two alpha helixes, there's two polymers of DNA, one here starting at 5' and moving down to the 3', and the other here, 3' moving down at the 5'. Two polymers that are being held together. We'll look more into that in a little while, but they're being held together by hydrogen bonds. Now, hydrogen bonds, we'll talk a little more about them later, but they're weak bonds in between molecules. The covalent bonds, represented by the lines here, the covalent bonds hold the molecule together. The hydrogen bonds of the molecule hold the two molecules together. So when we look at this molecule of DNA, this polymer of DNA, and then this polymer of DNA, they're being held together by these hydrogen bonds. Between T and A, there's two hydrogen bonds. Between G and C, there's 3 hydrogen bonds. That'll be important later when we discuss more about DNA. When you look at a DNA molecule, there's a backbone represented again by this blue line. The ladders in the DNA that you see here represent the nitrogenous bases here. This nitrogenous base, this nitrogenous base. Right? So when we look at this, this has a sugar phosphate backbone. This sugar phosphate backbone is called that because of the sugar and the phosphate that make up the structure. So when you look at this here, this sugar and phosphate, right, makes up a sugar-phosphate backbone. Again, this sugar and phosphate over here make up the sugar and phosphate on the other end. The two blue lines here are represented by represent this sugar-phosphate backbone. See the label of it here. The base pairing is this nitrogenous base being paired with this nitrogenous base. Why are they called nitrogenous bases? Because they have nitrogen in them. You have the two ring, the double ring, and the single ring. Adenine and guanine are double ring structures. Cytosine and thymine are single ring structures. Here's a better drawing of the nitrogenous base. A closer look at it. Here's the ribose sugar, 1, 2, 3, 4, and 5. And then the phosphate, sugar-phosphate backbone. Sugar-phosphate backbone. All right, so I hope when you look at the sugar-phosphate backbones, you see here and here and here, that again are represented by this blue line here. You can also identify that there are nitrogenous bases in between them that are held together. This holds together this strand of DNA to this strand of DNA by these hydrogen bonds. So this strand of DNA and this strand of DNA, they're held together by these hydrogen bonds in between. This is important because that means that you can separate the two strands without breaking apart the actual polymer itself. Since there's two polymers here being held together by hydrogen bonds, if you break the hydrogen bonds, you can still keep the polymer together. The order of the letters, T, G, A, T, T, what have you, can stay constant while separating the two strands. You'll see why that's important later. They're parallel in the sense that one strand is next to the other. Now they're also an alpha helix twisting pattern, but they run parallel to each other. They're anti-parallel because you'll see this one side is 3', the other side's 5'. They're opposite one another, so they're anti-parallel. Anti-parallel, and they complement each other. They have complementary strands. One strand complements the other, meaning they don't match exactly. If you see a T here, it's not going to be a T over here. It's going to be an A. T and A are always matching, where G and C are always matching. You're never going to see a T with a C or a G with an A. They're always matched, one across from the other, A and T, G and C. This again, these are all important points. Let's move on to the next slide then. So when we're looking at the different nitrogenous bases, there are purines, and the purines are represented by the double ring structure. And you have pyrimidines, and the pyrimidines are represented by single ring structures. The purines and pyrimidines go with each other, so that's why the A matches the T and the G the C, because a pyrimidine goes with a purine and a purine goes with a pyrimidine. So A goes with T and G goes with C. That's how they match up. Anytime you see a G here, on the other side you're going to see a C. Anytime you see an A here, on the other side you're going to see a T. That goes all the way up and down this DNA molecule. RNA is only single-stranded. Where DNA is double-stranded. 2 polymers. 1, 2. They both have sugar phosphate backbones. Notice they both have sugar phosphate backbones. Of course, DNA has deoxyribo and ribose, where RNA has ribonucleic acid, or ribose sugar. Another big difference is where you have, in DNA, you have a T. In RNA, you have a U. So that U replaces a T. You'll see here, this is not a T, it's a U. Now when they match, the U always matches with an A, and the C still matches with a G, and the G still matches with the C, and the A though this time matches with a U. So there will be matching, and you'll see that you'll see it when we do it. But when it's in its native form, when it's not being created, RNA is single-stranded. It does form a temporary double strand with DNA when it's being created. Let's go on. So this drawing represents that anti-parallel notion. Here's your 5' end. Here's your 3' end. On the opposite side, there's your 3' end. There's your 5' end. Now, what is that 5' and 3' again? Remembering that this is the 0. You have to, imagine turning the molecule on its side. This would be the first carbon. This is the second here. Here's your third, your fourth, and there's your 5th carbon, 5'. On that 5' end, there's a phosphate. This is your 5' end phosphate. Here's your 0 here. That's your 1, 2, 3, 4, 5'. Of course, on your 3', there's a hydroxyl group. Don't forget, you see the hydroxyl group here. Remembering that hydroxyl is OH. Remember that hydroxyl is equal to OH, right? And remembering that phosphate is PO4, which would be 4 oxygens. Right, you see that here. This is your phosphate group. This is a hydroxyl group. There's a hydroxyl group. It's always on the 3' end, and there's always a phosphate on the 5' end. All right. Now, what is your nucleotide again? Remembering that your monomer of DNA, this is the polymer of DNA. But if you remember which one is your monomer, of course, with every polymer you have a monomer. A bunch of these all linked together. You could designate that monomer by highlighting it or circling it. If we did that, we could circle the monomer of DNA this way. Be phosphate, a sugar molecule. Let me make this a little smaller. So phosphate, a sugar molecule, and the nitrogenous base. That would be one monomer. That's monomer 1. Then you have a second monomer, a phosphate, sugar, and another nitrogenous base. That's the second. And third, phosphate, sugar, nitrogenous base. Fourth, phosphate, sugar, nitrogenous base. In order, in opposite order, anti-parallel to the other complementary strand. So again, complementary, where you see A, over here you're going to see T. Where you see, over here you see G, here you'll see C. G and C. Then A and T. So this would be A, this would be T. This is A. So T you see here, there'd be A here. If this is A, this is T. If this is G, this is G. Over here is C. If this is C, over here is G. So they're always complementing each other. C complements G, A complements T. If this were RNA matching with DNA, and will happen during transcription, that where you see A, you'll see a U on RNA. Where you see a T on DNA, you'll see an A on RNA. So if you see a U anywhere along the polymer, you know that that's a polymer of RNA. If you see a T, then you know it's a polymer of DNA. All right. So those are some clues as to which molecule is which. So when you're looking at DNA, you're looking at a chromosome. So when we look at DNAs, we're thinking, you need to be thinking in a eukaryote, it'll be single strand, it'll be linear, there'll be a 5' end, there'll be a 3' end. These are copies of each other, so there's two sister chromatids here connected together. So this is one copy of the DNA molecule, and there's another copy of the DNA molecule. Now when they say DNA molecules here, each of these DNA molecules are actually considered, technically there's two molecules here, two polymers, one from 5' to 3', the other from 3' to 5', wound together in a double helix, all wound together really tightly into this thing we call a chromosome. So one copy, all your genes are found here. All the information that makes you who you are found here. And because these things are anti-parallel, because they have a 5' end and a 3' end, they have direction. So when I'm reading these letters, A, C, T, G, C, T, A, T, T, what have you, there's a direction I can read it at. And in fact, your cells and the biochemistry of how your cells work actually read it from 5' to 3'. So you're looking at, you're reading it, 5', you keep reading. Yeah, that looks like this would be 3', 3' here. Reading it straight down, it would be read the other way, and you'd read the letters as if it were a book. So this is actually a code that translates into proteins that functions that do things, that create traits. So if you will, this is what you may have learned in middle school as your genetic code or your genotype. Your phenotype is what is produced by this code. This code is a sequence of nucleotides. There's an order that's read in a direction. So let's talk about the differences then between DNA and RNA. What do we know about DNA? Let's summarize it. First, we know about DNA that there is a sugar phosphate backbone. That sugar phosphate backbone has a deoxyribose. It's represented by the letters DNA, which stand for deoxyribonucleic acid. So far, we know that much about DNA. We know more about DNA now. I hope you've learned more about DNA. The second thing I hope you learned about DNA is that DNA also has a nitrogenous bases. These nitrogenous bases have purines and pyrimidines. The purines are guanine and adenine. The pyrimidines are thymine and cytosine. So that seems like a lot, but it's not all we've learned. We've also learned that the monomer of DNA, the DNA monomer, is known as a nucleotide. And that nucleotide has, in its most basic form, a pentose sugar, who has an oxygen at the center. From the one, it has a nitrogenous base. From the first carbon is a nitrogenous base that comes off of it. And that nitrogenous base is either a purine or pyrimidine. It could be either one. From the third carbon comes a hydroxyl group. It's a 3 prime. And on the 5 prime, there is a phosphate attached. So these 3 components form the nucleotide: the phosphate, the sugar, and the nitrogenous base. And that means that this is a monomer. So we can take these monomers, as we've drawn out before, this phosphate, sugar, and nitrogenous base, and connect it to another monomer, phosphate, sugar, and nitrogenous base, over and over and over, going on for billions of nucleotides that make up your DNA molecules in your cells. So we know there's nitrogenous bases, we know there's purines and pyrimidines, we know that the monomer of DNA is made of phosphate, nitrogenous base, and sugar. The sugar's deoxyribose nucleic acid. We know there's a backbone to the DNA molecule, is represented by this blue section here, that, if you'll see, it highlights the sugar and the phosphate because it's a sugar-phosphate backbone. We know also some other things about how these 2 molecules are connected together. We know, for instance, that these 2 polymers of DNA, these 2 strands, right? So DNA is double-stranded. And that we know that they're a helix. They twist around. We know now that the helix twists because of the geometry of the molecules. So double-stranded helix. We know that hydrogen bonds hold these 2 strands together. The hydrogen bonds are guanine and cytosine, and that's a triple bond, triple hydrogen bond. Remembering that hydrogen bonds are weaker than covalent bonds, and that they hold molecules to other molecules. They don't hold the molecule together. What holds the molecules together? Covalent bonds. Those are very strong. But the bonds are weaker, and that's a good thing. These hydrogen bonds are weaker. They're being held together by weaker forces. That's a good thing because that allows the cell, allows life to separate the molecules the strands. I'm going to use strands, the DNA strands. Without breaking the polymer. So I can take these two, this strand, this polymer, and this polymer here, and I can separate them by breaking the hydrogen bonds without breaking up the covalent bonds that hold the polymer together. So this sequence, this DNA sequence, this code can stay together, and so can this one, even though I separate them. That way I can read them. In order to read what's inside here, I have to be able to open them up, unzip the ladder, if you will. So we know a lot about DNA. Oh, we know that it's anti-parallel. Let's not forget that. So we know that there's a direction to DNA. It's 5' to 3'. And we know that there's that one strand, that's strand 1, is anti-parallel to strand 2, which also has a direction. So this strand is going to match this strand. 5' matches with 3, and 3 to 5. You don't see it here, but you saw it in the previous slides. So that's a lot. We know a lot about DNA. We learned a lot about DNA in a couple of days. But RNA, it's a very similar molecule. Oh, yeah, we already said ATGC. We know that RNA does have a sugar phosphate backbone, so this is the same. Except that you have a ribose sugar. It has that oxygen on the second carbon. It has nitrogenous bases. RNA has nitrogenous bases as well. But the interesting thing about these nitrogenous bases has purines and pyrimidines. But RNA has G and A, the two purines. That's fine. The pyrimidines actually end up being U and C. Remembering that A is going to match up with U and G with C, right? But an interesting change that RNA has no T in it. So if you see a molecule, a polymer that has T in it, you know it's DNA. If it has a U in it, you know it's RNA. 3, the monomers are the same. 4, it's single-stranded. RNA is single-stranded. 5, it does have direction. It has the same direction as DNA. In other words, you have a 5' and then it goes to a 3'. There's no complementary strand because it's single-stranded. However, it will match up with DNA when in the making of RNA, you will have to match up the DNA to its RNA nucleotide. You'll see what I mean when we cover transcription. So that's really the summary of everything we've learned about RNA and DNA, all starting with that initial observation that this sugar, this carbohydrate, along with a phosphate, is the backbone of this DNA molecule, and that a nitrogenous base form the ladders or rungs in this ladder. If you see, look at this as a ladder, the rungs, the two sides that are attracted to each other that hold this polymer to this polymer. The rungs are the nitrogenous bases, which in DNA are GNA, T and C, and in RNA it's GNA, U and C. Remembering that in RNA you have a U, where in DNA you have a T. Alright, so that takes care of what we discussed. It summarizes what we've discussed in class today. I hope this helped. Have a good day.